Okay, well thanks very much for the introduction and for the invitation to come here and speak to you today. As Joe says, I'm supposed to have retired last year. You might want to talk to my wife about how effective that's been. I'm still keeping quite busy. And one of the things that I do, having left UK STEMCELL Bank and hPSC and the MHRA, is to work more one of my primary activities is as director for the International Stem Cell Banking Initiative, along with Andreas Kurtz, who you already heard from, and Rosie Asazi from University of Miami, who used to run the International Ethics Working Party. So, before I move on, I thought it might be worth just looking back and thinking about when we're talking about standards, what do we actually mean by standards? And there are a whole range of different things which people call standards. And there's a sort of hierarchy if you look at this. Firstly, the laws, the regulation on ethics and medical products, particularly in relation to this discussion for cell therapies and product safety testing. And under those, regulatory guidance, which sort of provides you with something that's like the highway code in the UK that tells you how to meet the regulation or guides you on how to meet the regulation. But under that, one of the things that this activity that I've been involved in has been particularly connected with is development of best practice. And particularly where that comes from a consensus from expert groups. This can be very powerful and the regulators can be very, very responsive to it. There are generic cell culture standards, some of which have included stem cell applications, such as the publication in the British Journal of Cancer from Geraghty et al. In 2014. And earlier than that, there was one developed which came out of the toxicology area, where they were interested in standardizing in vitro toxicology assays. And there were a lot of problems with contaminated, misidentified, wrong, incorrect cell lines. And so guidance came out from that group called Good Cell Culture Practice in 2005, and that's now under revision and now includes things like 3D culture systems, organoids, cells on a chip, and also human pluripotent stem cell culture as well. So these guidances, there are now guidances that are beginning to address this, and I think the toxicology area has been particularly important in driving this from an industry perspective. And OECD also has come out with guidance on in vitro assay development, which links into these good practice documents. There are also protocols, pharmacopoeia methods, and there are a whole raft of international standards organisation standards now coming in, which were being directly developed for regenerative medicines and cell therapies in particular. There are also the protocol-based journals, and all of these provide us with helpful protocols, ways of doing things, sometimes which are very specific and required for industrial testing, and others which are more generic. However good the protocols are, the way people do them is very often different. And that's one of the reasons that the institute that I used to work for, the National Institute for Biological Standards and Control, actually exists, because people are not always able to make the same things or do the same things in different places. So it's really important to have this final point, the physical reference materials, which help people to compare biological materials in different centres at different times, or even the same manufacturer at different times. And that's been really crucial in the history of developing biological products in medicines, in gradually improving the quality and activity of those materials. There are a number of reference material manufacturing centres. We're lucky in the UK we have the NIBSC that I mentioned, which has been doing this in biological medicines for many years, and there are others, such as IRMM, that deal with different areas. These are also connected with international bodies, such as the Expert Committee on Biological Standardization, run by WHO, and the JCTLM, which operate to develop high-level international standards. And of course, there are also metrology centres, and we're lucky in the UK, we have a number of these, NIBSC, the National Physics Lab, and the Laboratory of the Government Chemist. And outside of the UK, obviously, and Europe, there are other organisations, NIST and ASTM in the US, and a number of others, the Standards Coordinating Body, etc. So a lot of activity and structures to take us along in standardisation. I think the main area that I'm going to be concentrating on today is in the middle part, on the best practice side of things. So at the core of providing reliable cell cultures of any type, whether they're stem cells or anything else, is a robust banking process to provide cells to users of the cells at the similar cell age over a period of time, so you're not constantly getting slightly different cells. Also a robust quality control program, A range of issues have been discussed for stem cells, and I'll talk a little bit about some of that work in a moment. And the ones in red are really the ones where there are already established standards, ISO standards and other standards that we can use in the stem cell area. And the area of identity testing is really where I started out on this with cell cultures some 30 years ago. And we're still trying to improve the standards that relate to use of authentic cells. With stem cell lines in particular, and this is a slide courtesy of UK Stem Cell Bank, you can follow a stem cell culture even over just a series of successive passages, and the levels of stem cell markers that you see in those cultures can vary quite considerably, as you can see with this OCT4 staining in flow cytometry analysis ranging from 40 to 70%. So it could be really helpful in these cases to have reference materials, physical reference materials that enable you to compare one batch of cells with another. Also, looking at genome status, this is an area obviously it's going to be discussed later today, and a really challenging area for developing a standard method. One of the important things I think coming from my perspective is how do you apply tests that will give you a reliable result that's repeatedly of a repeated standard each time you do it on different cell banks, and can you do that in a reasonable time frame at a reasonable cost and with the right sensitivities. And very often, if you're looking at genetic stability, obviously the first thing that you would think about is probably whole genome sequencing. But in terms of a quality control assay, unfortunately, that very often asks more questions than it answers for you, and it's very difficult to get a specific result that says pass or fail, because it's a grey area. So I think most stem cell banks these days are using the traditional karyological tests. And also array SNPs is becoming very popular as a routine method of quality control. The other area I thought I'd mention very briefly, and it's already been discussed quite very thoroughly by Peter Andrews, is the developing a standard pluripotency assay. As you heard, there are various methodologies available, but none is ideal or tell you that the cell line will generate a specific cell type. So there can be a lot of confusion by people who say, look, your cell line passed this pluripotency assay, but we've now got that cell line and we can't make X or Y cell line. And I've heard a number of cases where this has occurred. So we probably need new assays, perhaps along the lines of the ones that Professor Pei talked about earlier, looking at the chromatin, have some kind of screening assay that we can apply. The other thing that is quite worrying to me, and I know a number of people in the audience have spoken to this as well in the past, and this is the use of the term pluripotency markers. Now, if anybody has found one, they're going to be rich people. I don't know of one. If you do find one, please let me know. But these generally tend to get even in the literature, used as labels for things like the stage-specific early antigen markers, the Trafalgar antigen markers, and the self-renewal SOX2 and OCT4 markers as being pluripotency markers. And clearly, they are either self-renewal or stem cell-associated markers, but they also appear on a range of other non-pluripotent stem cells. So they are necessary, but probably not sufficient to demonstrate a pluripotent stem cell. The other thing I thought I'd mention, which is something that's been quite a powerful force in this area of standardization for pluripotent stem cells in particular, is the International Stem Cell Forum. That was initiated by George Roeder back in 2004, who brought together a number of international funders to help support projects that could only be done on an international basis. One of the major ones was the one led by Peter Andrews, who spoke earlier, and now Martin Perra, who is also in the audience and speak later. And that project brought together up to 30-odd labs in different countries to collaborate to establish what are the standard markers for human ES cells in the early stages, comparing feeder-free media, a lot of confusion about which was the best method to use, genetic stability and what was happening in these cell cultures, and then pluripotency assays, which should actually be 2018, sorry, the error there. And then also looking at genetic change and how that relates to safety in more recent discussions. There's also an ISCF ethics working party led by Barbara Knoppers and Rosie Asazi from Canada, now in the US, which dealt with a number of issues specific to iPS cells and ES cells, which affect the original donors. The International Stem Cell Bank Initiative is the project which I started up at the UK Stem Cell Bank in 2007, along with colleagues from around the world, some of which you will have heard from today. And this is an activity which engages a very diverse range of expertise and knowledge, both in stem cell banks, but also leading stem cell research groups, including those in the ISCI project, scientific societies, regulators, industry funders, and policy groups, all of which have an interest in supplying good quality cells and making sure researchers get the right material. It involves now over 24 countries. There are regular workshops on key issues for pluripotent stem cell banking, and then we try to publish useful consensus that comes out of those discussions, and there are a number of people in the audience here who have been to those meetings and supported those discussions. Probably one of the most recent, the most relevant recent publications from that group is the considerations for the development of seed stocks of pluripotent stem cells for clinical application, and that is really soup to nuts from donor selection through the cell banking process, quality control, storage and shipment, and the special issues that we need to think about for pluripotent stem cells, also as considerations of what we mean by pluripotency, tumorigenicity, etc., and the safety issues. Just before I finish, I thought I'd just mention the Human Pluripotent Stem Cell Registry. You've already heard a description of that from my colleague Andreas Kurtz, so I won't spend any time on that. Other than to say it's now got this, I feel a very important point now, that it's now directly linked to cell banks such as YCell. UK Stem Cell Bank has cell lines on here. The EBIS collection has, I think, around 500 cell lines now registered through this process. And hPSC has become a core part of that EBIS IPS cell bank for disease studies. So an increasingly important activity, which is closely connected with the ISCBI activities. So in conclusion, I think there are two different kinds of standard that are going to be important for pluripotent stem cells. Firstly, consensus on best practice, and there are a number of groups that I've spoken about who have been working to support this and develop it. And I think this is going to be an ongoing process. You don't just write one of these things and it works for the next 20 years. We know with GCCP that's got to be renewed every 5 to 10 years minimum. And then the physical standards, and some of these are now under development at my old institute, National Institute for Biological Standards and Control, and both Ola O'Shea and Professor Jack Price from there are present in the audience if you want to hear more about that. There are also scientific challenges which I've reflected on. I think the key ones are the genetic stability issue, what assays should we be using, and pluripotency assays, how can we accurately predict what the cell lines will make, and that's still, as we heard earlier, not quite there. Then I haven't mentioned this really, it's quite a complex area, standardisation on ethics. A number of groups have tried to establish standard consent forms and continue to do so, but it's always struggles with local arrangements for assuring consent and national differences. A second part of this is now increasingly important, that's the EU General Data Protection Regulations, which are now here. They came in May of this year and could be a bit of an issue for international exchange of materials and data, been discussed at the ICBI meetings. So finally, I'd just like to say that stem cell biobanks, working with the scientific community, I think can play a really important part, significant role in promoting standardisation in this field, and I look forward to another few years of facilitating this through the ICBI. Thanks very much.